everyone. Trisha here. We're going to go over cellular respiration today, so go ahead and break out those worksheets. This is personally one of my favorite portions of biology, so hopefully you enjoy it too, and if not, hopefully, my hope is that by the end of this worksheet it'll be making a little bit more sense, because cellular respiration is a big concept to wrap your mind around. So let's go ahead and switch my screen over to the actual worksheet, which is this guy right here. It says right up on top, how can cells convert the energy in glucose to ATP? And so what we're going to go over is this summary formula right here. C6H12O6 plus 6O2 turns into 6H2O, and 6CO2, plus 6H2O plus energy. So you're going to have glucose over on the left with 6 oxygen, and that transforms into 6 carbon dioxide and 6 water and energy. Energy being this guy here, adenosine triphosphate, also known as ATP. So as we go through the worksheet, we'll actually be answering what's happening in this big graph up here. So let's break out some pens or pencils or markers or erasable pens, or in my case, this lovely pen over here, this Apple Pencil. We'll start answering each one of these questions here. So first one is asking, where is each of the reactants in the overall process? So this right here tells you what the reactants are. C6, which is your glucose, and then you have your O2, which is the other reactant. So you're going to put down here, not these two, because I mean they are the reactants in the equation. It's in stoichiometric formula over here. But what you're really saying is, where exactly do they fall in this whole process? Is it oxidation phosphorylation? Is it actually the electron transport chain, citric acid cycle? Well, it says it right here. Glycolysis is glucose to pyruvate. So really, the C6H12 and the CO2, yeah, they have your reactants here, but they're asking where do you see that? You get glucose and CO2, and O2. You get the glucose, which is this molecule right here. That is going to be your glycolysis. And the best way to think about it is glycogen, glucose, glycolysis. That's, bio tries to make it easy for you. Let's just put it this way. Let it try and be obvious. So now your O2 is whereabouts in this whole cycle. Is it glycolysis, pyruvate, citric acid cycle? Where it's going to be primarily those 6O2 is going to be in the oxidative phosphorylation itself. So you're going to come on down here and you're going to write down oxidative, and then hopefully my writing is not atrocious. Please in the chat let me know if my writing is atrocious. Wait, I didn't want to do this in cursive. Pardon-moi. Now your your 6 CO2 and your 6 H2O, which are your products up here. So here is one product, here is another product, and here is your energy. We'll just make a big thing around it, energy. You want to look at this and be like, where exactly is that going to be? Well, your 6CO2 is one of the products that's actually coming, a little bit of OChem around here, but I promise it's not scary. Where it's coming from is, this is connected to that. So your glycolysis is part of creating this 6CO2. So where do you see the CO2? You see it here in pyruvate because glucose turns into pyruvate in glycolysis. Then you also see it in what is called your acetyl-CoA, which is short for your acetyl coenzyme A. That's what the CoA stands for, coenzyme A. And the last part that you see it is in the citric acid cycle, which also comes with another name that it also goes by, which is called the Krebs cycle. Okay, so that's where you see it. Basically, it means like, wow, that produces a lot of CO2. It does. And in that process of CO2 creation, it's making lots and lots of ATP, aka that last portion right here of energy. So, in the 6CO2, you're going to write down your pyruvate. You're going to say acetyl CoA. And the last one is going to be the Krebs / citric acid cycle. Again, Krebs and citric acid cycle is the same thing. Okay? So on that note, where do you find your energy? Your energy is the byproduct. So here's your yellow, right? Your energy is the byproduct of basically everything in this entire thing. So this is energy, this is energy, this is energy. ATP is energy. Adenosine triphosphate is energy. So your energy, again, byproduct, it's a product from those reactants that you had earlier. Your energy is going to be seen in ATP. Your energy is going to be also seen in your Krebs cycle. You're also going to see it in glycolysis as well. And why do you say that? Because here's glycolysis and there's ATP. So in the process of glycolysis, ATP is created. And the last portion where you'll see it is in the oxidative phosphorylation. Forgetting about a little product here, water. Water is also a byproduct from your chemical reaction. So you started with your reactants. I'm going to make the reactants underlined in purple. And then I'm going to have over here in blue your products. We're just missing one last one. Where are we going to find some H2O? And really where you'll find it the most is in oxidative phosphorylation, in the chemiosmosis electron transport chain. Okay? So you'll go ahead and write down here, oxidative phosphorylation. And feel free as I'm going through this, ask any questions that you have about this stuff. You will be seeing this throughout, especially if you're pre-med. This whole thing right here is a gigantic portion of the biochem section. You don't have to know what's the word I'm looking for? You have to know everything. It's not like, oh, you just don't have to know this, or you don't have to know that. You literally have to know everything on a gigantic level, as in it's like that minute. You need to know it down to this like tiny level. And I'll even demonstrate how small you have to know. I'm just going to go back to energy, and I'm just going to add a little bit more data in terms of how much energy is produced in every single one of these little cycles here, okay? So ATP is energy. Inside the Krebs cycle, you're going to create what is called, and I'm just going to make a little arrow over here. It's going to be called 2-GTP, which is a different form of energy, okay? And then you also have a little arrow right here. In oxidative phosphorylation, you have about up to 34 ATP. That is a lot of ATP, like a lot. So it is working really hard to create energy inside of the cell for your body. And then, oh yes, and then glycolysis, you have about 2 ATP that pop out, which again, you can see it right here. You get about 2 of these that pop out as you go from glycolysis, and then it goes through cytosol and then creates that ATP. And if you're wondering what GTP stands for, I'm just pulling out my old biochem notes really quick, just in case you want to know. Your GTP is your, if you just wanted to know, GTP also stands for your guanosine triphosphate. You don't need to know that. And if you're wondering what the guanosine comes from, if you go into genetics, it is, actually you'll learn it this quarter really, because you're going to get the basics of genetics in this term. Towards your final, you have what is called your DNA strand and then your RNA strand. And in the DNA strand, you have adenosine, guanine, hold on. Adenosine, thymine, and then thymine turns into uracil, and then cytosine is in the DNA phase, guanine is in both. So guanine is where this comes from. It's one of our building blocks of life, chemical building blocks of life. So that was a lot of time spent for such a small thing, so we're going to try and get through the rest of this, I swear. So now, what is the overall function of glycolysis? The overall function of glycolysis is basically to produce about 2-pyruvate and then generates 2 ATP and 2 NADH. And then your 2 ATP and the 2 NADH and the 2 ATP are generated per glucose. And I'll move that down a little bit so that you have room to write up more up here. And then your 2-pyruvate come from oxidized glucose. Okay? And the overall function of the Krebs cycle, you're going to notice something. There is a theme here. It's oxidation. Oxidative phosphorylation is the big thing. Oxidation happens a lot in this cellular respiration process. So first word's going to be oxidation of pyruvate / acetyl coenzyme A to CO2. Okay? It also is going to generate those 2 guanosine triphosphates, 6 NADH, and 2 FADH2 per, well, it just looks like a Z. Bad writing me. Per glucose. And then what is the overall function? If you're noticing a theme, it is oxidation of NADH and FADH to H2O. It also does that for NAD, or FAD. So if you're noticing here, you had an H here and then you lost an H here, okay? That's a little bit of some acid-base chemistry. So for my Gen Chem people, you're going to see that way later in Gen Chem 3. If you've already been through Gen Chem 3 and you have not, or you're currently taking OChem, this is pretty much essential to understanding why in OChem we have to know what is a proton donator, aka an H, right here, an H plus proton donator and an H plus proton acceptor, because then it's going to determine acidity. And this is part of why it's important to know how this all works, because then it explains the pH of the human body, pH of parts of our system, where like pH is lower in our stomach acid versus pH of the rest of our like actual like blood. It differs in different areas of our system for this reason. And then the way that our bodies are set up is done in a way where if something is a pH of 1.5 here in our tummies, it is set up in a way where it should not technically leak into elsewhere of the human body. But sometimes things happen and then it happens and then you're like, oops, oh my goodness. So that is one. And then the other thing we'll write down here for what is the overall function is that we're going to write down, it generates, again, what I was saying, generates that H plus, so proton, that H plus ion, concentration gradient. To form ATP. And that area, if you're just wondering, is right at the end of this very area, right here in oxidative phosphorylation. So oxidative phosphorylation, you have your electron transport chain, and it literally is, when you look at it, like from, I believe there's a slide on it class, if you look at it very, very closely, it almost looks like it's just going up and down, like almost like a wave, where you have FADH turns into FAD plus, and then you have NADH turn into NAD plus, and then all of those H's are basically floating around on the outside, and then they slam through what is called that ATP synthase, and through ATP synthase you have your ADP over here with a hanging out on the side kind of phosphate, and it's through it passing through, and the H is coming through that ATP synthase that literally looks like, you know, one of those centrifuges that are out in like if you've seen The Martian, it looks like a centrifuge, but like really, really, really fastly sped up. It's really cool to look at, like on a cellular level, and it spits through all of this energy, and that is what transforms that ADP into ATP that the body needs to basically do a lot of stuff, like have our muscles work and actually do motions like this. So I just think that's really cool. But I'm a nerd, so. I think there might have been a little bit of like a formatting error for this section. So I recreated the table on my end, and what you can do is like move this or just kind of like follow it down and write and create your own table using this as a base, and fill it in and do your best. You can, if you're scanning it in, you can just write it on the back of the page. So they're asking here, are the compounds listed here, which I don't have any yet because I'm going to write them down with this guy right here. Hello. They're asking if something is used or something is produced in these three different portions of cellular respiration: glycolysis, Krebs cycle, or oxidative phosphorylation. So what you're going to write on the side over here are the following words. You're going to write down glucose, O2, CO2, H2O. Sorry, you can even see that. My apologies. H2O, ATP, ADP with that phosphate. So when I was talking about ADP to P, that and the P is kind of like hanging off to the side. Hello, I'm here. That's what ADP plus the P with the little I stands for. Okay, and then you have your NAD, H, and finally NAD plus. And then I'm going to change my color to just a nice light-ish blue. So glycolysis. Is it used in glucose? Glycolysis, glucose? Yep, it is used. O2? Nope. CO2? Not really. H2O? No. And you know that now because of what we went through much earlier when we were trying to figure out where is what actually positioned in terms of reactants and products in this formula up here. Okay? So none of that. ATP is produced, and again, you know that because, hi, there's ATP and it came back from glycolysis itself. So we'll go ahead and write that down, that it is produced. Produced. In terms of the ADP to P, it is produced because you have to, in order to create ATP, you have to have ADP. So if it helps me to say adenosine triphosphate versus adenosine diphosphate, let me know, because sometimes just saying the initials is really confusing. So this is a produced, because it's produced in glycolysis and used. So it's both produced and used, because you have to use the adenosine diphosphate to create the adenosine triphosphate. So NADH is going to be produced. And again, that is because it's going from the glycolysis into pyruvate, where it is then sent into that Krebs cycle. Okay? And finally, the last thing is used. And as I was saying earlier, knowing when you take away and add a hydrogen proton is basically how this is all balanced out. And you are producing the NADH to then go through to the Krebs cycle. But the only way you're going to get there is you have the NADH used and then an H is basically attached on, if that makes sense. So it's used to then produce something. Now, Krebs cycle, we'll switch that over to red. O2 is 1000% used in the Krebs cycle. CO2 is what is produced. We don't really use water, but we do have a produced. Oops. And again, it's that guanosine triphosphate that is produced. This is used, your adenosine diphosphate. This is also used, NADH. So NADH is not just in one area, it's in a couple of areas. And the last thing here is that NADH is produced. Last one, let's switch to the color green. Let's go green. For oxidative phosphorylation. Well, around this point, you have to use a little bit of glucose. You got to use some of that sugars to be able to actually energize oxidative phosphorylation. You don't have any O2, you don't have any CO2 because that was basically used up and produced in the Krebs cycle. But you know what happens now? Because you used and produced O2 and CO2, you now have an H2O and that is produced. This right here is also produced, your ATP, which again, gone through that, it's like one of the biggest byproducts that happen at the very end of oxidative phosphorylation. ADP is used, which again I described before. You have to use ADP to create the ATP. And in order to do that, you have to basically depronate the NADH to then spit out the H's to then have them come back through the ATP synthase to then produce that ATP. And finally, NADH is produced. Excellent. Let us move on. Switching back to a black color. The cell supply of ADP, P, and NADH is finite, limited. What happens to cellular respiration when all of the cells NADH has been converted to NAD plus has been converted to NADH? Basically, what happens in the shortest way possible, easiest way to explain it is the cell cannot conduct any process involving NAD plus to NADH conversion. It's because, in case you want to know, it's because glycolysis, sorry for my bad writing, y'all, and Krebs cycle produce NADH. And without it, so I'm going to basically move that up a little bit, and without it, the process is shut down. Sound good, everyone? All right, let's move on. Now from here, this is asking if the Krebs cycle does not require oxygen, why does cellular respiration stop after glycolysis when no oxygen is present? Basically, it stops because of one major thing, and I feel like I'm harping on it a lot because it's kind of important. Without oxygen, there's no oxidative phosphorylation. And it's because it's literally in the name. Oxidative oxygen, no oxidative phosphorylation. So that's the answer to that question. And by the way, if you want me to go back and have you kind of review this, just let me know, say something in the chat, and I'll go ahead and do that. Okay? So why does it stop? And it is because, and I'm going to go ahead and just initial this as, let's go ahead and write it all out. Oxidative phosphorylation cannot occur. So that's the major one. The other major thing is that NADH cannot be oxidized. And then the last thing is that pyruvate, which is the thing that glycolysis happens. You have glucose that turns into pyruvate. Just if you're wondering, wait, did she even bring that up? Yes. So pyruvate is the thing that occurs during glycolysis. So you have glucose turn into pyruvate. Pyruvate goes through an oxidation process that turns into that acetyl-CoA. Literally as it says right here on the tin. Hello, tin. We go ahead and go back down. All the way down here. So now the pyruvate basically cannot convert to, I can't spell, acetyl-CoA. So this is why it's very, very important to have oxygen. Because even though the Krebs cycle doesn't require it, everything else does. No oxidative phosphorylation, NADH cannot be oxidized, and as we just described, NADH is in a lot of this. A lot of this is happening with NADH. And the last thing is pyruvate cannot convert to that acetyl coenzyme A, which is essential for the entire Krebs cycle. Okay? Next one. Now, this one's asking, many organisms can withstand periods of oxygen debt, aka anaerobic conditions. Yeast undergoing oxygen debt converts pyruvic acid to ethanol and carbon dioxide. Animals undergoing oxygen debt convert pyruvic acid to lactic acid. Pyruvic acid is fairly non-toxic in even high concentrations. Both ethanol and lactic acid are toxic even moderate concentrations. Explain why this conversion occurs to organisms. It basically comes back to NAD and NADH. For this one, we focus more on NAD plus. So NAD plus isn't available. Let's just say it isn't here. Therefore, glycolysis stops. No ATP is produced. And the cell dies, aka apoptosis. Just a nice fancy word. Bam, throwing it at you. Okay? This is known as fermentation. Which, if you've heard the word fermentation, maybe you might have heard it in a couple of different things, but for me, I've heard it when it comes to beer making. Now, how efficient is that fermentation? How efficient is cellular respiration? Remember that efficiency is the amount of useful energy as ATP gained during the process divided by the total amount of energy available in glucose. Use 686 kilocalories as the total energy available in 1 mole of glucose and 8 kilocalories as the energy available in 1 mole of ATP. What? Not to worry. We're going to do the math together. It's probably one of the most mathiest questions I've seen in a while. But I'll break it down for you. So let's go ahead and have fermentation in the color of red. And I'll go ahead and make this nice and big so you guys can follow along. So let's do 8 calories. And I like to write it this way: per mole ATP. And you're going to times that. Oops. I'm going to put that here. With 2 ATP. Oops. Over 1. And that's going to give you your 16 calories. And if you're wondering how I got that, it's because the ATP cancels out. Down here, you're going to take that 16 calories. Oops, let's go back to red. Take those 16 calories. And then we're going to say here is that you're going to say for every 686 kilocalories, you're going to have 1 mole glucose. For my people who are in Gen Chem, this is probably not the happiest thing in the world to look at right now. And if you were in Gen Chem and you didn't like it, probably still not the happiest thing in the world to look at right now. And then what you're going to say up here is that for every, let's see here, you have 1 mole glucose. And then for every 1 mole glucose, you're going to have about 2 mole ATP. And again, if you're wondering why, it's because mole glucose, calorie. That's how it cancels out. And that will give you an answer, which you'll then times it by 100 to then give you their final answer of 2.3%. Lots of math. I know. We'll move the purple over for aerobic respiration. Similar setup, 8 calories over 1. And then you put over here that you're going to have about, oh, wait, I forgot. Oh, wait, no, I can put that back. Calories per 1 mole ATP. And then up here, you're going to say for every 1 kilocal, you have 1 mole ATP. Well, not 1 mole, it's 38, excuse me. 38 mole ATP, which then turns into your 340 kilocal. Then you bring that down here, 304 kilocal. And that you have down here, 686 kilocal per 1 mole glucose. And then bring this out. And then you know you're going to be timesing that by 100 to give you your percentage. So then from here, you'll go ahead and say for every 1 mole glucose, we're going to have about 38 mole ATP. If you're wondering why such a big number, it's a gigantic number because that's the conversion factor for aerobic respiration. Because, going back up, that is a lot of ATP that is generated from oxidative phosphorylation, right? Now 34 plus 2 plus 2 gives you what? Gave you 38 mole ATP. That's where it comes from. Now you do all of the math, and then you're going to come up with your answer of 44.3%. And there it is. Math done. Let us move on, yes? Yes. So let's just double check how we're doing right now. Okay, we're almost done. I'd say this is about the halfway point. Hopefully you're hanging on in there. You guys are more than welcome to chat through and ask questions. It's like, don't make me feel like I'm alone out here, please. Anyway, I'm making myself laugh. Moving on. Let's get this guy done. So why can't cells store large quantities of ATP? Hint, consider both the chemical stability of the molecule and the cell's osmotic potential. So probably an initial thing to think about is, well, that's a lot of energy. Because again, if that's how much it is for aerobic respiration, wow, we really cannot store that much because it'll probably just go and explode, and that would be a bad thing. But that's not what to really write about for this section. The thing you want to think about is, well, basically ATP is how big. Is it a big molecule or a small molecule? So one thing is that ATP is a small molecule because, it only it's like hydrogen's a really small proton. So the thing that it creates is also pretty much going to be small. But that's not the biggest factor here. It's more about, because it's so small, it can put so much energy with it. Again, this is kind of going back to some Gen Chem stuff. So if you've taken Gen Chem, this might sound familiar, where the smaller of a atomic radius the atom is, the faster it's going to be and the more energy it's going to have. So it's probably going to be a little bit electronegative. And that if it's bigger, so if it's going that, so if this over here is, well, I'll just mirror this. This is the halogen group over here, and then over here, well, this would be your halogen group. This would be your alkali metal, alkali earth metals. Here is where you have the fastest ones, and then over here you have the slowest ones. The only one who's faster than all of them is hydrogen yonder up here, and that's why it's number one. It's the fastest one because it's the smallest one. So it's a small molecule that is highly reactive. And by reactivity, I mean that speed, that energy. Okay? Which is why it's hard to store it in a cell. Or hence why it's hard to store in a cell. And then the other thing you want to talk about is the osmotic potential, because again, think about the cell's osmotic potential with osmotic potential high ATP concentration in a cell would make a cell hypertonic to fluid, and therefore make the cell burst. So what I said earlier wasn't wrong. It's just that it wasn't enough, if that makes sense. Because just to say, well, if I put it all in there and in large quantities, it would probably explode. I mean, I wasn't wrong. But, and I just had a spam bot. Yay! Fantastic. I don't know why I got a spam bot. Fantastic. I don't know how to delete that. I'm clearly an old woman. I will delete that later. But basically, as I was saying, it is really, fine to have said what I said to begin with, which is the cell was going to burst because it's just too much in there. So, boop. But gave it a little bit more and say because it is a small molecule, due to what we kind of remember a little bit about Gen Chem, it's going to be reactive, which means it's going to have a lot of energy. And if we have a lot of it concentrated into a cell, it's going to change that osmotic pressure, that osmotic potential inside of that cell, make it hypertonic, which means it's going to want, give me fluid, give me fluid, give me fluid, and then it's too much, and then boop, bye-bye cell. Okay? So now we can move on to B, which is given that cells can't store ATP for long periods of time, how do they store energy? And that's why we have fats. They store energy in fats, oils, or starches. And it's because on a very, very chemistry level, if you look at how fats are structured on a cell, I don't want to say a cellular, but if you look at what a fat looks like on a chemical level, and what an oil looks like in its structure, and how the bonds are interwoven, then you can see okay, they are strong enough to hold ATP for a long period of time because of how, is it polar or non-polar? Probably because it's going to be a little bit, see, it's going to be non-polar most likely because ATP will probably be polar. And it's basically like oil and water. They don't mix. They're going to stay away from each other. It's a heterogeneous mixture, not a homogeneous mixture, which is kind of what you want when it comes to cells. That would be bad if it was homogeneous. So then what are the advantages of storing energy in this alternate forms? Basically, no effect on that osmotic pressure. Potential is the biggest one, I would say, like pretty much the biggest one. And again, like I said before, that on a chemical level, chemically more stable. So this would be number one, and here, chemically more stable. And then we can move on. So here is number 9. To make a 5 molarity solution of hydrochloric acid, we add 400 milliliters of 12.5 molarity hydrochloric acid. That's a lot of hydrochloric acid. To 600 milliliters of distilled water. For adding the acid, however, we place the flask containing the distilled water into the sink because the solution can heat up so rapidly that the flask breaks. How is this reaction similar to what happens in chemiosmosis? How is it different? Well, let's try and just break it down pretty simple. Let's do some similarities here in, let's do green. We haven't done that one in a while. Similarities in green. In both, we add acid to water, generate differences in concentration gradient and energy released as forms of heat. Perfect. Now for the differences. Let's do that one in a darker blue. Differences. Energy is controlled as these H proton ions pass through ATP synthase. And then some energy, which again for my Gen Chem students is probably going to sound pretty familiar. Some energy is released as heat. Oops. Sometimes I just end up writing cursive. And then just a little caveat here, some energy is released as heat. Some is captured in chemical bonds of ATP. All right, moving on. Woo! This is a big one. Okay. Let's just break it down pretty quickly. If you got 1,000 grams of glucose to grow 10 grams of an anaerobic bacterium, how many grams of glucose would it take to grow 10 grams of that same bacterium if it was respirating aerobically? Well, I'm not going to read the rest of that. Let's just get right to the point of it. Yes. So, you already know from way earlier that aerobic respiration produces about 38 ATP per glucose molecule. Okay? And you already know that anaerobic respiration produces about 2 ATP. And I'm just going to do this as a fast per molecule, or glucose molecule. So then what do you do next? So you're like, okay, well, if I have this much and I have this much, how do I figure out if I'm going to be multiplying, if I'm going to be dividing? Basically, there's some math that I don't think is necessary for this right now. How you want to think about it is, if I have 38 of this and then I have 2 of this, what do I need in order to make this more efficient at X amount of grams of anaerobic bacterium? Basically, think about this way. Oh, I guess I'll do the math. You're going to take about 1,000 grams glucose, and then you're going to divide it here. You move this down here, 100 grams of glucose, and then you're going to divide that by about 19. And the reason why it's 19 is because it is 19 times more efficient with this type of aerobic respiration. With aerobic respiration. And then your math down here is you're going to have 1,000 grams of glucose divided by 19. And that is going to equal about 50 grams of glucose. Why did I spell it like that? Also, oh, it's not 19. I'm sorry. Ignore me. Why did I say the word 19? Hi. It's 20 times more efficient. Good job, me. So then you have your 20 down here. And then you have your 50 grams of glucose. I'm sorry, my fault. Required. Now that makes more sense. All right. One more page to go, and we are then going to be done. So, next one is all about mitochondria. Isolated from liver cells, can be used to study the rate of electron transport in response to the variety of chemicals. The rate of electron transport is measured as the rate of disappearance of O2 from the solution using an oxygen-sensitive electrode. How can we justify using the disappearance of oxygen from the solution as a measure of electron transport? This sounds really complicated. It's pretty darn simple. All you got to do is just literally write down the formula, which is your C6H12O6 plus 6O2 and that turns into your 6CO2 plus 6H2O plus energy. That's all you have to write down for that one. This one is a lot of words, but essentially it's asking, since human beings require about 2,000 calories of energy per day, and that for each mole of glucose oxidized, about 686 kilocalories of energy is released, then why don't humans spontaneously combust? And that is a really good question. And essentially what it's going to come down to is the fact that during cellular respiration, in particularly the oxidation of glucose. So the thing that we have to think is glucose oxidation. And what does that do? The glucose oxidation basically is going to, what's the word I'm looking for? So during the cellular respiration, the glucose oxidation's energy produced not all released at once. It's done in phases. And those phases are, of course, glycolysis and pyruvate oxidation, Krebs cycle, electron transport chain. So that's what we have to think. Basically what our cells do all the time. We got 1, 2, 3, 4 more portions. Almost done. If at any point you need to just bounce, it's okay, but I'm going to try and get through all of this. And really, it's actually, yeah, we're just about done, I swear. So this one is going to be asking, how does the requirement for a high NAD plus NADH ratio support caloric restriction hypothesis? Because it's saying here that a gene was recently identified that encodes a protein that increases longevity in mice, which is awesome, cool. Maybe that'll translate to human beings. Basically, it's asking what is the ratio for a high NAD plus NADH ratio, okay? So what you're going to say here is that less NADH, H, produced by glycolysis and Krebs cycle will increase NAD plus NADH ratio. Bam! Awesome. This one is asking an active college age student, wow, college age athlete, can burn more than 3,000 calories per day in exercise. If conversion of 1 mole to ATP to ADP produces that much calories, well then how many moles of ATP need to be produced per day for this energy to be met? I'll do the math really quickly. All you're going to write down here is 3,000 calories per day. And that, actually, let me just do that. We'll make it even quicker. Per day divided by your 7.3 cal per mole. ATP produces 421 mole ATP. All right, we made it. Last little portion in the last minute. So now, if the molecular weight of ATP is 573, how much would the required ATP weigh in kilograms? Again, a little bit of some math here. We're going to take that 411 mole of ATP, and you're going to times it per 573 grams. Oops, that's bad work. Grams per mole. And that's going to go ahead and turn into about a gigantic, huge number of grams, which you can then convert to 235 kilograms. And basically, to explain these results, pretty simple. ATP breaks down to adenosine, diphosphate with that PI. And it's continuously recycled. To ATP during cellular respiration. And that is it, everyone. Woohoo! Just a minute over. All good. Yahoo! You made it. Great job, everyone. Awesome. All right, any questions about any of this? It's 7 o'clock. I'm sure we're tired, exhausted, but it's done, everyone. If you want, I'll just go ahead and scroll up again one last time. Just kind of go through a little bit. Yeah, if you've got any questions, feel free to, message me now. You can email me, whatever feels best to you. I can put in the chat what my email is if you want. I'm going to go ahead and stop the recording right now. So for those who are watching on YouTube, I'm going to go ahead and stop, and I hope you have a wonderful rest of your day.